this column, and this will experience lesser magnetic field than the third one, and the first one will experience the least magnetic field. Okay, is this clear to all of you? So when you give your magnetic field gx side to side, your omega naught, this is frequency encoding, this was phase encoding, and this is frequency encoding. Omega naught becomes omega 1. This entire column omega naught now becomes omega 1. The second column, can you tell me which one here, omega 1, omega 2, omega 3, omega 4, which one has the highest precessional frequency, omega 1 or 2 or 3 or 4, and why? Who can tell me?
Omega 4, because we are increasing from left to the right.
Right, very good. So that means omega 4. Here we are not changing any phase because we are
Phase.
not like, unlike GY was given, then again it was switched off. We are not doing that. GX is gradient is played only when you are receiving the signal. Okay.
Okay.
What is the unique thing you are noticing here? Phase is not remaining changed. Phase remains the same what you had done after phase encoding. What has changed is your frequency have changed. What do you find here? Can anybody tell me an observation?
Phase is same along the x-axis.
Yeah, phase has not changed. When you give GX, only frequency has changed. But what is the uniqueness after this complete? I've done a
Each each square will have a unique frequency and phase.
right,
Like
very good. Each voxel, we'll call it a
voxel,
voxel,
voxel.
which is represented as a pixel. Each voxel is characterized by a unique combination of frequency and phase. Imagine it takes us so much of effort to understand, and the physicists who have come up with this theory, because of which today we have the MRI. Okay, so this
Yes.
is after. Now it is only theory. You read this, you'll understand. X gradient is called frequency encoding. Why so? Because only frequency part changes, phase does not change. It's also called a readout gradient. Why readout gradient? Because it is sent when you are receiving the MR signal. Let us say B increases from left to right, exactly what we presume. But the protons in each vertical column experience the same magnetic field precise with same frequency. Protons in left hand column precess more slowly, those on the right hand precess faster. We have done all this corresponding frequency from left to right. Emitted frequencies will vary from lower to higher from the left side to the right side of the body. We have done it all. Same thing. Increasing B means increasing frequency, same thing. Now, you may ask that why do we do a GX gradient? Why didn't we stop at GY gradient? That is this part. What do you find here? What is the frequency here? Anybody? This is post GY. What is the frequency?
Post DY frequency remains constant.
Yeah, and that is omega naught, right?
Yeah.
So you will get one frequency like this. One frequency. Only a single frequency is received. That is the omega naught. If you don't do a GX, but if you pass it from side to side, then with each column you will identify by a unique frequency and you will get a spectrum. Frequency encoding thus creates proton columns, each of which have an identical frequency. Columns, because this fellow is going like this. Here it was coming like this, so it was creating rows. Okay, here it is going like this, so it is creating columns. with the fastest precession here, third fastest here, second fastest, and least here. So now that we have a slice, which is whenever we talk of the word slice, it means we have selected it with GZ gradient. Now that we have MR signal emitted from a slice, it consists of spectrum of phases plus frequencies. Now you have information, two variables, you have frequency and you have phase. You have to calculate both of them. Okay. Now suppose I have told this to you yesterday also. The first let us this 2D Fourier transform. The computer never sits that okay, first let me calculate the frequency, then I'll calculate the phase. This is done at the same time. So imagine how powerful the processor has to be. So we say that in MRI there is 2D Fourier transform. Why 2D? One dimension will measure the phase, the other dimension will measure the frequency. Now, suppose I have a 256 by 256 matrix. Instead of this 4 by 4, I have a 256 by 256 voxel. Then the pulse cycle is repeated how many times? As many rows are there. I told you, what is a pulse cycle? Can somebody tell me from the figures I had drawn? What is a pulse cycle? First, you have what? A 90-degree RF pulse. Then at TE by 2, at exactly half the time to echo, we put what? 180-degree RF pulse. At exactly TE, what do we get? We get our echo signal, time to echo. Then after some time, we again have something called TR, time to repeat. The second signal for the same slice, same slice. Again, when I am repeating at TR, I have my first 90-degree pulse. Again at TE by 2, I have my second 180-degree pulse. Same thing I am repeating, only at TR. Again, the third pulse echo sequence, I'll repeat the same thing. How many times will I repeat if there is a 4 by 4 matrix? 90 180 echo, 90 180 echo, 90 180 echo, 90 180 echo. 4 times I will repeat my pulse cycle if I have 4 by 4 matrix. If here I told you if you have 256 by 256 matrix, how many times will you repeat? 256 times. But what I have not drawn in the figures I have shown you earlier, I have shown you TR. Those of you didn't see this, I have shown TR in the figure. But at each TR, what are we doing? Anybody can say? This is very important to remember.
We are increasing the frequency a bit.
We are increasing the, not the frequency, Baba, wrong
The
terminology. We are increasing
phase,
the, anybody else?
the phase of the frequency.
No, you can't play around with phase and frequency. You are increasing what?
Gradient.
Gradient. Very good. Which gradient?
Uh, the gradient of the frequency, the field gradient, right? Am
Uh,
I?
See, gradient of frequency and all are redundant things. What is a gradient? Gradient is a
Magnetic gradient.
gradient is a changing magnetic field. If I say not only if I say there is a gradient of temperature, what does it mean? Gradient means maybe I'm changing it from minus 10 degrees Celsius, minus 9 degrees Celsius, 8 degrees Celsius. -7, -6, -5, +1 degree Celsius, +4, +5. So it's a gradient. So don't say grade. So you somebody said gradient of frequency. Oh, that makes no sense. A gradient is a change, gradual change. You draw gradient curves, gradient lines. So here you have three gradient coils where you will bring about a gradient. What will happen if I have a gradient gz or a gy? Finally, you know what it does? Finally, it affects the external magnetic field B and makes that a gradient. Okay. So what I'm trying to say is people now just answer this for me. I think GY is pretty easy. Suppose I think my classes were attended by all the medical physics students. Can you tell me a typical sequence of MR? What does it consist of? Tell me. What does a typical sequence, MR sequence consist of? Try at least. Right at the beginning, what do we have? Supratit, tell me. Right at the beginning, what do we have in a MR sequence? Tell me. I'll help you. There's a pulse, right? Try to recall that figure where we have the decay, T2 start decay, T2 decay. You'll have not understood anything. It's that bad a situation. Anybody can say what is Supratit? Are you there? I don't think. Anybody else here? Yash, can you tell me?
Uh, Ma'am,
Right at
uh,
right at the beginning, we have a pulse, right? What is the
a 90
pulse?
degree RF pulse,
Excellent, excellent. Then,
and at half T echo, we provide a 180-degree RF pulse,
excellent.
which,
And then we will
kind of,
have a echo after some time, which we
yeah,
call as where in between you have rephasing, dephasing, and all that, and then you have echo, and that point is called what? TE,
time of echo,
time
TE,
to echo, right? Then there is a little bit of a gap, and then you have another term. What is it called?
time of repetition. The next
Right.
pulse of
Yeah.
radio frequency is given,
Very good.
and again the process of dephasing and rephasing it
Right.
continues.
So again at TR you will have a 90 degree. Again the same thing. You will have at T by 2 you will have a 180 degree. Again you will have another time to echo. Right. Again after some time you will have the second TR. Right. Time to repeat. My question is Yash, if I have a 4 by 4 matrix, then how many times I have to do this?
Just 4 times more.
Yes, at least 4 times MR sequence has to be repeated. And each time, what special thing I have to do with one of the gradients?
Sorry, I didn't get the question. Ma'am.
Okay.
First, a GY gradient is given
You are
so
very
that
correct that for 4 by 4 matrix, we have to repeat the sequence for the same slice 4 times. If it's a
yes,
generally not 4, it's a 256 by 256 voxel. Then we have to repeat this sequence of 90 degree, 180 degree, TE and TR again there. We have to repeat it 256 times. My next
yes,
question to you is each time, 256 times, each time one of the gradients has to be played. Which one is it? One of the gradients. Anybody else knows each time which gradient has to be increased a little bit? Ramkrishna, can you tell me? Half the people have not.
Ma'am, is it the Y gradient?
Very good. Y gradient has to be increased. So if I'm repeating my MR sequence 256 times to map 256 by 256 cells, how many time, how much I have to give increment to the Y gradient? How many times?
We'll have 256 different frequencies.
So I have to give increment to the GY how many times?
256 times.
Perfect. Perfect. Is that clear to all of you? The first MR sequence, I use the gradient G naught. Second MR sequence, I use G naught plus 1. Little steeper I make it. Third MR sequence for the same slice, I use G naught plus 2. So it keeps on making it steeper, because then only you can fill up all the data. I'll come to something why you keep on increasing, we'll understand. So are you a little clear about this? You have to read it up. Okay. Now that's what is written here. Can you all see this? To map a 256 by 256 matrix, the whole pulse cycle repeated 256 times. Pulse cycle is nothing but 90 180 degree TE again TR, 90 180 degree TE again at TR. Each time phase encoding gradient or GY is increased a little. During these 256 repetitions of my 90 180 degree, if any tissue moves, there is a misregistration and the signals will be attributed to the wrong pixels. This is a motion artifact we can do later, but any kind of movement will appear it is not coming from the slice. So the frequency and phase together enable unique identification of each voxel. Last one, unique identification, the combination. So the MR signal measured contains two pieces of information. The frequency locates the signal along the x-axis and the phase distribution within each frequency. So basically the frequency is always taken and then it is decomposed into its component frequencies like we have done here. We are going to break up this frequency into component frequencies. And in the phase, you will come to know corresponding to provides information on the place of origin of the corresponding signal. Now things are becoming better. For each voxel, you will know this signal is corresponding to the which place of origin. Earlier, we were getting information, we didn't know the exact location. So I've told you already why we say 2D FT, because Fourier transform has to be applied twice, once to map the frequency, once to get information about the phase. To perform such calculations, 256 equations, 256 unknowns, that is the phase, frequency and all, you need an array processor. What they're trying to tell you is that the lot of data to be processed. So you need an array processor for MRI. A normal computer processor doesn't work. Summarizing this, we have 3 dimensions: slice selection, frequency encoding, phase encoding and frequency encoding. Yeah, I've already told you this. For basic pulse sequences, one time through the pulse sequence. What is a pulse sequence? Can anybody tell me? What does one time through the pulse sequence mean? Tell me. One time through the pulse sequence. We just now discussed it, I think. What is one time through the pulse sequence telling us? Tell me, Roshna, are you there? Can anybody tell me this? What does it mean? One time through the pulse sequence. What is a pulse sequence?